So now we will be going through topic 6.1, digestion. And just a little warning, the intention of these videos is to aid with course material. So don't just rely on these videos, use these videos to help. So first we have the introduction. So digestion is the process by which food is broken down into smaller and simpler molecules that can be absorbed and utilized by the body. It involves both mechanical and chemical processes. Mechanical digestion refers to the physical breakdown of food into smaller pieces through chewing, grinding, and mixing by muscular contractions. Chemical digestion involves the release of digestive enzymes and acids that break down complex molecules such as carbohydrates, proteins, and fats into simpler forms that can be absorbed by the body. The end goal of digestion is to extract nutrients from food and convert them into energy, growth, and repair for the body's cells and tissues. So first of all, we have the alimentary canal, also known as the digestive tract or gastrointestinal tract. It is a long tube that runs all the way through the body and is responsible for digestion and absorption of food. It is a crucial system in the human body, allowing us to break down food into nutrients that can be used for energy, growth, and repair. So the alimentary canal begins at the mouth and extends all the way to the anus. It consists of several organs, each playing a specific role in the digestion process. Here is a brief overview of the organs and their functions. So the mouth is where the whole digestive process begins. So this is where food is ingested and mechanically broken down by chewing. Saliva is also simultaneously released, which contains enzymes that begin the chemical breakdown of carbohydrates. The enzyme is amylase. And then there is the pharynx and the esophagus. So from the mouth, the food passes through the pharynx, and that is a shared pathway for both air and food. And then the food runs through the esophagus, and the esophagus is a muscular tube that transports food from the mouth to the stomach using coordinated muscle contractions called peristalsis. And I will go through peristalsis in more depth in later slides. And then there is the stomach. The stomach is a highly muscular organ that further breaks down food mechanically and chemically. It secretes gastric juices containing enzymes and hydrochloric acid, which help in the digestion of proteins. The stomach also mixes and churns the food, turning it into semi-liquid mass called chyme. And then we have the small intestine. So the small intestine is the longest part of the alimentary canal, and it is where most of the digestion and absorption of nutrients occur. So the small intestine receives secretions from the liver and pancreas, which aid in the breakdown of carbohydrates, proteins, and fats. The inner surface of the small intestines is lined with tiny finger-like projections called villi, which greatly increase the surface area available for absorption. We will go through the entire process and structure of the small intestine in much more detail in later slides. And then there is the large intestine, also in brackets the colon. So the large intestine is responsible for absorbing water, electrolytes like salts, and vitamins produced by beneficial bacteria that reside there. It also helps in the formation and storage of feces. The colon is divided into several parts: the ascending colon, transverse, and descending. And it's really easy to remember. The ascending is the part that goes up, so it's the part on the left. Transverse is the one that looks horizontal, and the descending is the one on the right where it goes down. So then there is the rectum and anus. The rectum serves as the temporary storage site for feces until they are expelled through the anus through defecation. So through the entire length of the alimentary canal, various digestive enzymes and hormones are released to facilitate the digestion and absorption of nutrients. The process involves mechanical breakdowns and muscular contractions, as well as chemical breakdowns. And then we have the second set of organs, which are accessory organs. And these are organs that play a vital role in the digestive process by aiding in digestion, but they are not directly contributing to digestion. These organs are responsible for producing and releasing digestive enzymes, fluids, and substances that assist in breaking down food and facilitating nutrient absorption. Here are some of the major accessory organs involved in digestion. So first we have the liver. The liver is the largest internal organ and has multiple functions in digestion. It produces bile, a substance stored in the gallbladder, which is essential for the breakdown of breakdown and absorption of fats. Bile helps emulsify fats, breaking them down into smaller droplets, which increase the efficiency of fat digestion by enzymes. The liver also detoxifies harmful substances, stores nutrients, and produces certain proteins necessary for blood clotting. And then we have the gallbladder. The gallbladder is a small pear-shaped organ located beneath the liver. Its primary function is to store and concentrate bile produced by the liver. When fat enters the small intestines, the gallbladder releases bile into the digestive tract through the bile duct, aiding in fat digestion. And then we have the pancreas. The pancreas is both an endocrine and exocrine gland, but its exocrine function is particularly important for digestion. It produces digestive enzymes that are released into the small intestines to break down carbohydrates, proteins, and fats. The pancreas also secretes the bicarbonate, a substance that helps neutralize the acidic chyme from the stomach as it enters the small intestines. And then there is the salivary gland. The salivary gland includes the parotid, submandibular, and sublingual glands. They produce saliva, which contains the enzyme amylase, which begins the digestion of carbohydrates, as also previously mentioned. Saliva moistens and lubricates food, facilitating in chewing, swallowing, and the formation of the bolus for easier digestion and movement through the digestive tract. These accessory organs work in coordination with the organs of the alimentary canal, such as the mouth, stomach, small intestines, and large intestines, to ensure the effective breakdown, digestion, and absorption of nutrients from food we consume. Their contribution are crucial for maintaining proper digestive function and overall health. And here is a more simplified diagram of the digestive tract. This is one that you will be more likely expected to draw during exams. So make sure you have all of these parts and label them. And unlike this diagram, make sure the labeling lines are straight. And then we go on to mechanical digestion. Mechanical digestion is an essential process in the overall digestion of food, which involves the physical breakdown of large food particles into smaller fragments. It primarily occurs in the mouth and stomach, where various mechanisms help to break down the food mechanically, increasing its surface area for further chemical digestion. The process of mechanical digestion begins in the mouth, with the act of chewing, also known as mastication. So teeth play a crucial role in this by cutting and grinding and crushing the food into smaller pieces. The tongue assists by manipulating the food and mixing it with saliva, which not only adds extra lubrication, but also contains digestive enzymes, amylase, that initiates the breakdown of carbohydrates. Once the food is sufficiently chewed and mixed with saliva, it forms into a small mass called a bolus, which is then swallowed and travels down the esophagus into the stomach. In the stomach, mechanical digestion continues through the action of muscular contractions. The muscular contractions contract and churn the food, mixing it with gastric juices that contain enzymes and acids. This vigorous mixing breaks down the food further into smaller particles, creating a semi-liquid mixture called the chyme. The chyme is then gradually released from the stomach into the small intestines, where the process of chemical digestion takes place. The mechanical digestion in the small intestine occurs through the coordinated contractions of its muscular walls, which propel the chyme along its length while thoroughly mixing it with digestive enzymes, bile, and other fluids. And I will get to this again on its own slide. And then mechanical digestion also involves the action of specialized structures in the digestive tract, such as teeth, already mentioned, which is like more broken down into types such as canines, which are specifically used for tearing and shredding, premolars and molars, which are used for crushing food. So a summary is that mechanical digestion prepares the food for chemical digestion by increasing its surface area and facilitating the act of enzymes and enhancing the absorption of nutrients. And now moving on to the movement of food. So peristalsis is a coordinated muscular movement that occurs in the digestive tract, specifically in the esophagus, stomach, and small intestines and large intestines. It plays a crucial role in propelling food with other substances through the digestive tract. Peristalsis is characterized by rhythmic contractions and relaxations of the smooth muscles that line the walls of the digestive organs. These muscular contractions create a wave-like motion that moves the contents of the digestive tract forward. This also explains why you are able to eat upside down and food sort of defies gravity and it still goes to your stomach. So in the esophagus, peristalsis helps to push the food bolus from the mouth to the stomach. As the food enters the esophagus, the muscles in the upper portion contract while the muscles below relax, creating a wave of contractions that push the food downwards. This process continues until the food reaches the stomach. In the stomach, peristaltic contractions mix the food with gastric juices, promoting mechanical digestion. These contractions also help to break down food into chyme. In the small intestine, peristalsis continues the movement of chyme along its length. The rhythmic contractions propel the chyme forward, allowing it to come in contact with absorptive surfaces of the small intestines, the villi and microvilli, for nutrient absorption. In the large intestine, peristalsis aids in the elimination of waste products. It helps to move the ingestible residue, including fiber, water, and bacteria, towards the rectum, where it is eventually eliminated as feces. Peristalsis is an involuntary process, controlled by the enetric nervous system, which is a network of nerves that regulate digestive system's functions. So peristalsis is a vital mechanism in the digestive system, enabling the movement of food and waste products throughout the gastrointestinal tract. And then we have segmentation. Segmentation is a digestive process that occurs primarily in the small intestine. It involves the contraction and relaxation of the circular muscles in the intestinal wall. In the intestinal wall. So unlike peristalsis, which propels the contents forward in one direction, segmentation involves localized contractions that squeeze and churn the chyme with the small intestine. These contractions occur at different points along the intestine, creating separate compartments or segments. The purpose of segmentation is to facilitate the thorough mixing of chyme with digestive enzymes, bile, and other fluids present in the small intestine. This mixing helps to ensure that the chyme is exposed to the absorptive surface of the intestine, allowing for optimal absorption. During segmentation, the circular muscles contract, constricting the intestine at that specific point. This constricted segment then undergoes rhythmic contractions, which alternate with relaxation of the surrounding segments. These contractions mix the chyme within that particular segment, ensuring that it comes in contact with the villi. As already mentioned, as the chyme is mixed, nutrients are more effectively broken down and absorbed by the intestinal wall. This process allows for more efficiency in extracting the nutrients. And segmentation also serves to slow down the movement of the chyme through the small intestine, which allows for it to be more absorbed by the villi. So yeah. And then we have chemical digestion, which is the process by which large complex molecules in food are broken down into smaller, simpler molecules through the action of enzymes and other chemicals. It primarily occurs in the stomach and small intestine. Enzymes secreted by the salivary glands, stomach, pancreas, and small intestine play a crucial role in chemical digestion by breaking down proteins, carbohydrates, and fats into their constituent building blocks. The breakdown enables the body to absorb and utilize these nutrients for energy production, growth, and maintenance of bodily functions. Chemical digestion complements mechanical digestion and is essential for the complete breakdown and absorption of nutrients. So first of all, we have stomach acids, most primarily hydrochloric acid, HCl, which plays a vital role in the process of digestion. The stomach lining contains specialized cells called perotial cells that secrete HCl in the gastric pits and other substances into the stomach cavity, which is the hole, like the area. So the main function of the stomach acid is to create an acidic environment for the stomach, which serves several important purposes. Firstly, the low pH, which is highly acidic, helps to activate the enzyme pepsinogen, which is secreted by the stomach's chief cells. Pepsinogen is converted into its active form, pepsin, in the presence of the acidic environment. Pepsin is responsible for the breakdown of proteins into smaller peptides. Secondly, the stomach acid helps kill or inhibit the growth of potentially harmful microorganisms that may be present in the food we consume. The acidic environment of the stomach creates an unfavorable condition for most bacteria, pathogens, which aids in the prevention of foodborne illnesses. Furthermore, stomach acid plays a role in the breakdown of certain food components. For example, it helps denature proteins, unfolding their complex structure and making them more accessible to enzymatic digestion. Lastly, stomach acid assists in the absorption of certain nutrients such as calcium, iron, vitamin B12. The acid in breaking down food particles and releasing these nutrients, allowing for their subsequent absorption in the small intestine. While stomach acid is essential for absorption, the stomach lining is protected from its corrosive effect by a layer of mucus. This mucus forms a protective barrier, preventing the acid from damaging the stomach tissue. And then we have bile, which is also a very important substance involved in digestion. And it is produced by the liver and stored in the gallbladder until it is needed. Bile is composed of bile salts, bile pigments, cholesterol, and other substances. Its main role is to aid in the emulsification and digestion of fats. Bile salts act as emulsifiers, breaking down large fat globules into smaller droplets, increasing their surface area and making them more accessible to the action of digestive enzymes. When food-containing fats enter the small intestine, the gallbladder contracts and releases bile into the duodenum, which is the first part of the small intestine, it is released into the common bile duct. Bile salts in the bile interact with the fat droplets, forming micelles, which further assist in the breakdown and dispersion of fats. The emulsified fats are then acted upon by pancreatic lipases, enzymes secreted by the pancreas, which break down the fats into fatty acids and glycerol. This process of emulsification and enzymatic digestion allow for the efficient absorption of fats in the small intestine. In addition to its role in the fat digestion, bile also aids in the absorption of fat-soluble vitamins such as vitamin A, D, E, and K, and helps in the excretion of waste products, particularly bilirubin, a pigment derived from the breakdown of red blood cells. This is not necessary to know, just a fun fact. After aiding in digestion, bile is reabsorbed in the small intestine and recycled back into the liver for further use. The recycling process helps conserve bile salts and ensures they are continuous and available for fat digestion. And then we have digestive enzymes. There are several, so let's get through this. They are specialized proteins that facilitate the breakdown of complex food molecules into smaller, simpler substances, allowing for their absorption and utilization by the body. So first we have amylase, which is an enzyme produced in the salivary glands and pancreas that breaks down carbohydrates, starch, and sugars into smaller molecules such as glucose. Then there's proteases, which includes pepsin and trypsin, and they are enzymes responsible for the digestion of proteins. Pepsin is produced in the stomach and begins the process of protein digestion by breaking them down into smaller peptide. Trypsin is secreted by the pancreas and further breaks down into peptides. Breakdowns the peptides into even smaller peptides and amino acids. And then there's lipases, and they're enzymes that break down fats into fatty acids and glycerol. They are produced by the pancreas and help in the digestion and absorption of dietary fats. Lactase is an enzyme produced by the small intestine that helps in the digestion of lactose, the sugar found in milk and dairy products. Lactase breaks down lactose into glucose and galactose, so it can be absorbed. Other enzymes like maltase, sucrase, and peptidases are involved in the further breakdown of carbohydrates, sugars, and peptides, respectively, into their simpler form. These digestive enzymes work in the specific pH environment and are activated at different stages of digestion. They act on the food within the gastrointestinal tract, breaking down complex molecules into smaller units that can be absorbed through intestinal lining into the bloodstream. The release of digestive enzymes is tightly regulated and coordinated to ensure optimal digestion and absorption of nutrients. Any deficiency or dysfunction in the production of activity of these enzymes can lead to digestive disorders and difficulty in nutrition absorption. Moving on to the small intestine. So in the process of digestion and nutrient absorption, it is the longest section of the digestive tract, measuring up to 6 meters in length. So the main function of the small intestine includes digestion, obviously. The small intestine is responsible for the further breakdown of food into smaller molecules through the action of enzymes. Enzymes from the small intestines, as well as those released by the pancreas, break down complex carbohydrates into simple sugars, proteins into amino acids, and fats into fatty acids and glycerol. Absorption and nutrients. The inner lining of the small intestine is highly specialized with numerous tiny finger-like projections called villi and microvilli, and these structures significantly increase the surface area available for absorption. The small intestine absorbs the breakdown product of carbohydrates, proteins, and fats, as well as vitamins, minerals, and water. These nutrients are then transported through the walls of the small intestine and into the bloodstream for distribution to the body's cells. And then there is absorption of water. The small intestine also absorbs water, which is essential for maintaining proper hydration and overall fluid balance in the body. And then there is secretion of digestive enzymes. The cells of the small intestine secrete various digestive enzymes, including maltase, sucrase, lactase, peptidase, and intestinal lipase, to further break down complex nutrients into their simplest form. And then there is movement and mixing. Peristaltic contractions and segmentation movements occur in the small intestine, and that helps with mixing of food and digestive enzymes. So overall, the small intestine is responsible for completing the digestive process initiated in the stomach and ensuring the absorption of nutrients into the bloodstream. And now we will get into the structure of the small intestine. The small intestine is a tubular organ that follows the stomach into the digestive tract. It consists of 3 main sections: the duodenum, jejunum, and ileum. Here's a brief description of the structure of the small intestine. So first we have the duodenum. The duodenum is the first and shortest segment of the small intestine. It receives partially digested food from the stomach, along with digestive enzymes and bile from the pancreas and gallbladder, respectively. The duodenum is C-shaped and connects to the stomach at the pyloric sphincter. Then there is the jejunum. The second part, the middle part, and the inner lining of the jejunum is characterized by numerous finger-like projections called villi, which greatly increase the surface area available for nutrient absorption. The jejunum is the primary site for the absorption of nutrients such as carbohydrates, proteins, fats, vitamins, and minerals. And then there is the ileum. The ileum is the final section of the small intestine, extending from the jejunum to the large intestine. Like the jejunum, the ileum is lined with the villi for nutrient absorption. It also has specialized lymphoid tissue known as Pear patches, which plays a role in immune defense. And then going on to the cross-sectional structure of the small intestines. So it has very distinct layers, as you can see in this diagram. So there is the mucosa. The innermost layer is the mucosa, and it contains the villi and microvilli. These structures increase the surface area for nutrient absorption. The mucosa also has glands that secrete digestive enzymes and mucus. And then there's the submucosa. The submucosa is a layer of connective tissue that contains blood vessels, lymphatic vessels, and nerves. It supplies nutrients to the intestinal wall and transports absorbed nutrients away. Then there's the muscularis. The muscularis is a smooth muscle layer responsible for peristaltic contractions and movement of food through the small intestine. Then there is the serosa, which is the outermost layer, which is a thin protective membrane that covers the small intestine and provides a slippery surface for movement within the abdominal cavity. The structure of the small intestine, with its extensive surface area and specialized features, is designed to maximize nutrient absorption and facilitation of digestion and processing of food. And moving on, we'll get into the structure of the villi, as it is extremely essential in absorption. So these are finger-like projections which are found on the inner lining of small intestine and greatly increase the surface area, enhancing the efficiency of absorption. And here are the main functions of the villi. So as previously mentioned, the main one is increasing surface area. The villi significantly expand the surface area of the small intestine. This increased surface area allows for greater contact between digested food and absorptive cells of the small intestine, maximizing the absorption. Then there's nutrient absorption. So each villus contains specialized cells that are responsible for absorbing nutrients, and they're called enterocytes, and they have microvilli, also known as brush border on their surface, which further increases the absorptive surface area. These cells absorb the broken down products of carbohydrates, protein, and fats, as well as vitamins, minerals, and water, and transport them directly into the bloodstream. And then we have transport mechanisms. The villi contain blood vessels and a network of lymphatic vessels called lacteals. These vessels facilitate the transport of absorbed nutrients. Nutrients such as glucose and amino acids are absorbed into the blood capillaries within the villi and transported to the liver via hepatic portal system. Fatty acids and fat-soluble vitamins are all absorbed into the lacteals and enter the lymphatic system before reaching the bloodstream. And then we have enzyme secretion. Cells within the villi secrete various digestive enzymes such as sucrase, lactase, maltase, and peptidases. These enzymes are involved in the further breakdown of carbohydrates, proteins, and peptides into their simplest form. Then there's the immune function. So the villi contain specialized immune cells and lymphoid tissue called Peyer's patches. These components help protect against potential pathogens that play a role in the body's immune defense with the intestinal tract. And so the structure of the villi in the intestines is specifically designed to maximize the surface area available for nutrients. So here is a diagram, and I will be explaining it. So this shape and location. Villi are like very small finger-like projections, and there are numerous amounts of them, and they extend into the lumen of the small intestines, and the lumen is the empty space inside. And they are present in the mucosal lining of both the jejunum and ileum, the two sections of the small intestine. So they have epithelial cells. The cells, so on the surface of each villus is several epithelial cells. It covers the surface. And specialized epithelial cells are known as enterocytes. Enterocytes are responsible for absorbing nutrients from the digestive food. Then there's microvilli. The enterocytes on the surface of villi possess even smaller microscopic projection called microvilli, and this further increases the surface area available for nutrient absorption. And then there is the capillary network. So within each villus, there is a dense network of blood capillaries. These capillaries are responsible for the absorption of water-soluble nutrients such as sugars, amino acids, and after absorption, these nutrients are transported through the capillary network to the liver via the hepatic portal system. And then there are lacteals. So in addition to the blood capillary, the villus also contains a lacteal, which is a specialized lymphatic capillary. Lacteals are involved in the absorption of dietary fats and fat-soluble vitamins. Fatty acids and fat-soluble vitamins are absorbed into the lacteals and directly enters the lymphatic system before eventually reaching the bloodstream. And the intricate structure of the villi with their extensive surface area, microvilli, capillaries, lacteals, and specialized cells allow for efficient absorption of nutrients during the process of digestion. And then moving on to membrane transport in the digestive system. So various methods of membrane transport are involved in the movement of nutrients and substances across the membrane of cells in the digestive system. And these transport mechanisms ensure the absorption of essential molecules from digested food into their bloodstream. So we will be going through different modes of membrane transport and some examples. So first of all, we have passive diffusion, and which consists of both osmosis and facilitated diffusion. So first we have passive diffusion. So passive diffusion is the simplest form of membrane transport that occurs down a concentration gradient. Small nonpolar molecules such as water, oxygen, and some lipids can passively diffuse through the cell membrane without need for ATP or transport proteins. And then there is facilitated diffusion, also does not need ATP, but facilitated diffusion involves the transport of larger molecules or charged particles across the cell membrane with the assistance of specific transport proteins. These proteins, called carrier proteins or channel proteins, facilitate the movement of substances such as glucose, amino acids, and ions across the membrane. And then we have active transport. So active transport is the process by molecules and ions which are transported against their concentration gradients. So this requires energy in the form of ATP. The transport mechanism is essential for the absorption of nutrients such as glucose, amino acids, and ions that may be present in lower concentrations in the intestinal lumen compared to the inside of the cell. And then we have two more types, endocytosis and exocytosis. So endocytosis is a process by which cells engulf and internalize large particles or macromolecules from the extracellular environment. In digestion, two types of endocytosis take place, phagocytosis and pinocytosis. Phagocytosis involves the engulfment of solid particles such as bacteria or cellular debris, while specialized cells called phagocytes. Pinocytosis, on the other hand, refers to the internalization of fluids and dissolved substances through the formation of small vesicles. And then we have exocytosis. Exocytosis is the reverse process of endocytosis, where substances are packaged into vesicles within the cell and then released outside the cell. In digestion, exocytosis is involved in the secretion of digestive enzymes, hormones, and other substances from cells into the digestive tract. And you can remember this by thinking of exiting when you think of exocytosis. So it's like exiting, and endocytosis is taking in. So these various methods of membrane transport work in conjunction to ensure the efficient absorption of nutrients, water, and other essential molecules. And yeah. And lastly, we have starch hydrolysis. So starch is a complex carbohydrate and one of the primary sources of energy in the human diet. It is composed of long chains of glucose molecules linked together. Starch is commonly found in foods such as grains, like wheat and rice, potatoes, and legumes. Starch hydrolysis refers to the process of breaking down starch into smaller and more digestible molecules, meaning that it is a catabolic reaction. The process is facilitated by the action of enzymes called amylase. There are two main types of amylase involved in starch hydrolysis. Salivary amylase, also known as ptyalin, is an enzyme produced in the salivary gland. When we chew food, salivary amylase is released into the mouth and begins the process of starch hydrolysis. It breaks down the long starch molecules into smaller fragments of maltose. And then there's pancreatic amylase. Pancreatic amylase is produced in the pancreas and is released into the small intestines, and it continues the hydrolysis of starch, further breaking down the maltose into smaller units. Pancreatic amylase converts them into maltose, maltotriose, and glucose. The hydrolysis of starch occurs in the digestive tract, primarily in the mouth and small intestines. The process involves the enzymatic cleavage of glycosidic bonds that hold the glucose molecules together in starch. As starch is hydrolyzed, the resulting smaller molecules are more easily and readily absorbed by the body. Once the starch is broken down into smaller units, the remaining steps of of carbohydrate digestion involves the action of specific enzymes in the small intestine. Enzymes such as maltase, sucrase, and lactase further break down the smaller molecules such as maltose, sucrose, and lactose into their individual glucose components. These glucose molecules can then be absorbed through the intestinal lining in the bloodstream for the distribution and utilization by body cells. And that is it for starch hydrolysis. And here is a little diagram more clearly showing the process. And then we have some paper 1 style questions to revise. So the first question is, what happens to starch in the small intestines? So you can think for yourself and then pause and then wait for the answers. And here is the answer. So amylase is secreted by the pancreas. Digests starch to enable absorption by the villi. And you can, there's more questions too. And there's this one. The diagram shows the layers of the tissue in a transverse section of the human small intestine. Which is the layer labeled with the letter X? It is the circular muscle. B. And then we have this question. Which organ in the human body secretes lipase, amylase, and protease? And of course the answer is A, the pancreas. And then the last question, glucose is absorbed through protein channels in the plasma membrane of epithelium cells in the small intestine. Which characteristics of glucose prevent its diffusion through the phospholipid bilayer? It is polar, therefore hydrophilic. It's important to remember that only nonpolar molecules can passively diffuse through the phospholipid bilayer.